So we've learned already a little bit about the two types of transport that exist in a cell or in an area, passive and active. We haven't really gone into detail on active at this point, and that's what this Vimeo was actually for. But just to review you, let's go through passive one more time. Passive transport does not require energy, no energy whatsoever, and molecules are all going to move from an area of high to low concentration. And the thing that is actually going to power the movement is that concentration gradient. So there's three actual types of transport that we've learned about that are passive transport. There's diffusion, osmosis, and facilitated diffusion. And so there's a diagram over here that kind of shows all those for those people like me that like pictures. And so you see in this situation all of these molecules, there's a higher concentration here to a lower concentration here. And this molecule right here, you'll see it moving across the membrane from high to low. So here's the I, because there's a whole bunch of that color dot, to low. And they're going across the membrane. They're not going through a protein, just through the phospholipid bilayer. And so this type of transport would be like if it was carbon dioxide or oxygen. And this is known as simple diffusion. So you'll see that explained right here in this little paragraph. So we talked about already that like iodine moving across the Ziploc baggie, or maybe if you had a balloon full of vanilla, you'd actually be able to smell the vanilla on the outside of the balloon. Candles in a room, somebody passing gas, perfume, all of those things are molecules that just move from one area to another because of concentration gradient. When you specifically are talking about water, and this is a water molecule here, moving across the membrane, it cannot move across these phospholipids because it is polar. And so it actually has to go through a doorway, and that doorway is called an aquaporin. And there are other things that are going to move across doorways like that if they're charged, like maybe some ions. But the specific movement of water is called osmosis. And then if we have something large, like maybe sugar or amino acids, and they need to come into the cell, they're going to need a specially shaped doorway. It's going to fit the kind of like an enzyme substrate. And in most situations, they are actually enzymes, and the substrate has to go through the doorway. But this is also facilitated diffusion, right here. And it doesn't require energy. And the reason it doesn't require energy is because they go down from a high to low concentration. So this is facilitated diffusion. So in order of advancement, like this is pretty easy, simple, that's what I was going for, all the way to the most complex of the structures and the types of passive transport. Alright, so let's review this paper that you should have on your left-hand page in your notebook. And so osmosis, does it require a protein? Yes. What's the name of that protein? An aquaporin. The molecules are going to move from high to low or low to high? High to low. Does it require energy? No. Is it active or passive? Passive. Okay, so you should be able to do this. Pause the Vimeo and go do this by yourself before I go through it. Alright, simple diffusion. Does it require a protein? No. How do the molecules move from high to low or low to high? Does it require energy? Is it active or passive? So you get the trend. Alright, facilitated diffusion. Does it require a protein? Yes. High to low or low to high? Does it require energy? Is it active or passive? I need to get her some food. Alright, back to transport. So we've got active transport, obviously is going to be the opposite of passive transport. Active people, they use a lot of energy when they're active. They don't just sit on the couch. So think of active requires energy. And the reason it's going to require energy to get things into the cell is because they're going to be humongous structures, or there's already a bunch of that particular molecule inside the cell. And so they're going to go against the gradient. And so, for example, like in this diagram, hopefully I've got it right here. You've got a lot of people on a train. Obviously, the concentration of people is higher on the train than off. And so naturally, it would be easy for them to just step off and move from high to low concentration. But look at these people up here in the top. To get on that train, to get into the train, there was already a bunch of people there. And to get on that train, don't you think it required a lot of energy? And so that would be moving from a low to a high concentration. And that happens in the cell as well. And so there's 3 different times when the cell is going to require energy to bring things into it. The first one is if you have something large that needs to be brought in. So this looks like maybe a bacteria or something, and it's being brought into the cell. And you already know your root words. Endo means into, cyto means cell, and osis is the condition of. So it's the condition of something being brought into the cell. So something large being engulfed into the cell. So if this was a white blood cell, for example, and this white blood cell was surrounding this bacteria to eat it, you'll see that the cell membrane right here actually moves around the structure. So that is going to require a lot of ATP. Now as the membrane moves around the structure, it surrounds it completely. And so you see this structure right here, how it's got a membrane around it? So this little structure right here is called a vesicle. And that vesicle has a membrane around it. So as that is digested, as the lysosomes release the enzymes, and as it gets broken down into smaller pieces, and the things that are kept, like if there were amino acids in here, they could be sent to the rough ER and to the ribosomes to be assembled into proteins, or maybe this little bacteria had some fat that could be broken down and utilized by the cell. But the waste that's left behind is going to need to be excreted. And because it's in a vesicle and it's pretty big compared to the cell, this vesicle will go up to the edge of the membrane and it will actually excrete the waste particles out. And that too will require energy. And that is called exocytosis, which means out, the condition of going out of the cell. And so I know this is kind of graphic, but you've probably seen people who've gone to the bathroom, or little babies especially, when they're going to the bathroom and things are coming out of their body. They're making like a poo-poo face because they're squeezing their face to make things go out when they're going potty. So think of the fact that it requires energy sometimes for people to excrete waste. The cell is the same way. Sorry, it's graphic in nature. Likewise, it's going to require, like the hot dog guy, Kobayashi, whenever he engulfs hot dog after hot dog, it's going to require him energy in order to do that. So I know that's kind of graphic, but maybe you'll remember it. I don't think I can actually draw a hot dog very well, so I'm going to just stop drawing a hot dog. Okay? Now, there are actually 2 types of endocytosis when things are brought into the cell. If they are things that are already broken down, I'm not talking about sugars and amino acids, a little bit bigger than that, broken down particles that come into the cell, that is the cell, it's called cell drinking. And sometimes you think of it as water, but it's not always water, because obviously water is the movement through those aquaporins. But if the cell is actually engulfing something that's already pretty small but bigger than a protein, that's called pinocytosis. Now, I want you to add the rest of this cell over here onto this diagram and give it a nucleus. So would that be prokaryotic or eukaryotic? Eukaryotic. And so this is an amoeba. And an amoeba is a single-celled organism you would probably catch if you drank the water in Mexico. Or you can get them in local ponds too. And they go in, there's several different types. Some of them eat your brain. And some of them just give you very bad diarrhea depending upon what kind they are. But this amoeba likes to engulf smaller little guys, usually like paramecium and things like that. And so because he's eating a whole entire particle, that's called phagocytosis, because phago means to eat. So another example of cells that like to eat are macrophages. Macrophages are big eaters, and that is actually the name of a white blood cell in your body whose job it is to eat viruses and bacteria. So macrophages actually eat their food through phagocytosis. We talked about earlier in the unit where it's believed that at one point a prokaryotic cell actually engulfed another prokaryotic cell and didn't digest it. Well, it did that through phagocytosis. And we talked about that with the endosymbiotic theory. So back to this practice sheet again. See if you can fill it out. Pause the video and see if you can fill it out. So, does exocytosis use a protein? No, it uses the whole membrane. Does it require energy? Yes. Is it active or passive? Well, yes means it's active. Endocytosis, bringing things into the cell. Does it use a protein? No. Does it require energy? Yes. And is it active or passive? Yes, active. All right.
The sodium-potassium pump is an active transport mechanism that is driven by the...
All right, so this is boring voice guy, and I recorded it once, and it's just way too boring for me to show you. But basically, the third type of transport is the sodium-potassium pump. And Gracie has started talking again, of course. And so what you have here, obviously, is a membrane right here, and you have an integral protein here. And you'll notice, kind of like with facilitated diffusion, that the protein has these sites, like an active site, like an enzyme does. And so what you can't see from this diagram is that there's a high concentration of sodium on the inside and potassium on the outside. So they're actually going to go against the gradient. And so the first thing that'll happen, because remember we're going against the gradient, there's already a lot of sodium there. We're going to put more sodium out there where there's already a large amount. And so 3 sodiums actually will bind to the protein. ATP is going to come in, and because this is the type of active transport, so ATP is going to be required to actually get that sodium to go where there's already a bunch of sodium. And so watch ATP right here. What happens? So ATP lost the phosphate group, and that's how energy is released. And that was enough energy to power the protein to change shape. So the sodium are going to pass through the doorway to the other side of the membrane. Now, whenever it does that, I'm going to speed it up a little bit. Now that the door's open, now we have a different molecule that can come in and fit in this particular active site, and that would be potassium. And really, and you should know that sodium and potassium are not round and triangular. They do have they're elements with a nucleus and electrons, and so they're fitting into a shape, yes, but I mean they're not obviously triangular and circular. And so 2 sodiums are going to be able to come back through the protein channel in order to get into the membrane. And those are also going against the gradient. And so the sodium-potassium pump is going to be huge with neurons, but for now we just need you to know that when the channel is open, you're moving against the gradient, and that 3 sodium will go in, ATP will go in and cause the door to be opened. Sodiums will go where there's already a bunch of sodium, and then potassium will come back through, 2 ions, okay, will come back through and go into the membrane. Now I said ions, and so you'll notice that both this K and this sodium both have a plus. Well, that tells you there's an ion. That means they have a charge because they've lost or gained an electron. In this case, they've lost an electron. They are less negative, so they're positive. Bingo. So in your notes, you'll see, like you just watch the animation, so you'll see that the sodium-potassium pump is the last example of active transport because we're going against. We're going where there's already a whole bunch of something from low to high. So we're going to need ATP, just like we talked about earlier. And so here's the membrane again, like you saw in that diagram, and the protein channel is open to the inside of the cell, and here's the outside. And you'll see that 3 sodium can go into the protein channel. ATP will power the protein to change shape and turn back into ADP because it lost the phosphate. Then the sodium ions will go outside of the cell. At the same time, the 2 potassium ions can come into the cell, and that doesn't necessarily require ATP to go back in. So then the potassium are going to come into the cell. And so what we're doing is we're increasing the concentration even more of sodium ions in one location and potassium in another. Okay? So the final part of this data table, just to review, is the sodium-potassium pump. And the sodium-potassium pump, did it use the protein? Yes. How did the molecules move? Was it low to high or high to low? Did it require energy? And therefore it is what type of transport? Okay? So as a result of doing these notes, you should be able to rock and roll and fill this in by yourself and know how they're different, how they're the same. Because I could easily say to you, what do simple diffusion and osmosis have in common? And you would say molecules that move down a gradient. Or I could say, what do facilitated diffusion and the sodium-potassium pump have in common? And you should say something about them needing a protein in order to transport those molecules. So think about those kind of things, because that's like memorizing this chart is just level 1, but being able to compare things and contrast them, that brings you up to level 2 thinking, and that's where we want you to be, if not at level 3.